Auzubillahiminash-shaytanir-rajim. Bismillahir-Rahmanir-Rahim. Dear students, Assalamu alaikum. Today my lecture is about another population of microorganism in the soil. You can say microorganism because these are also too small to be visible with naked eye. These are known as nematodes. Nematodes belong to a separate phylum of kingdom Animalia. As you can see, these are basically worms. These are round segmented worms, and they are active and they can move. They need moisture to move, feed, and reproduce. Under unfavorable conditions, nematodes produce inactive or dormant stages. Either it's a complete cessation of metabolism, that is known as cryptobiosis, or some dormant juvenile stages are produced. The reproduction in nematodes is either asexual or sexual, as in case of microorganism. And these nematodes produce a number of stages in their life cycles. There are 4 juvenile stages that have been observed between eggs and the adult stages. So nematodes are divided in different groups based on their physiology and morphology. These groups are basically based on their mouth parts, and also there are considerable metabolic differences among different members of these groups. Basically, the morphology of mouth parts are different for the different groups. For example, this is a bacterial feeder. This one is a fungal feeder. You see their mouth parts are different, and also their anatomy is also you can see is different. And then this is root feeder, or you can say plant eater, and then these are predatory, and then omnivores, and so on. So we can divide the nematodes into different groups based on their feed, what they eat. For example, plant, fungi, bacteria. Then organic matter. The ones that eat organic matter, we call them detritus eater. Then fauna, then eukaryotic cells eater, and so on. Omnivores are those groups which can feed on different types. They can feed on plant as well as animals. Now, first is we see a little bit detail about plant parasitic nematodes. These are important, and a number of people have done considerable amount of work on these kind of nematodes, because they cause important economic losses by causing diseases in plants. They are plant herbivores, and in this way they act as grazers. in the food chain. They also have an important role in below ground nutrient cycle. Then the other group is detritus nematodes. They are also very important as far as agriculture is concerned. They degrade the soil organic matter, and also they have a very important effect on soil structure because of their burrowing effect in the soil. Then they are used in composting process, and they increase the fertility of soil. So the compost in which the nematodes are used is known as vermicompost. And if I so this compost is basically a material that is made from different types of dead organic matter, and that is partially degraded material, and is used to increase fertility of the soil. Now, when we discuss about the role of nematodes in the soil systems, as you see, nematodes have a direct effect on soil microbiomes, as they feed on microorganisms, and also they have some other role that we'll be discussing in the next slides. So they have a role on regulation, and also they modify the size of soil microorganisms. And in this way, they accelerate the turnover of microbial biomass. They have a role in decomposition of soil organic matter, as we have already discussed, and they also increase fertility of soil because their excreta contains partially mineralized nutrients. And they have a role in moving microorganisms from one place to another place. Such transport is also known as phoretic transport. So the microorganisms are either carried over on nematode bodies, or they directly excrete the viable microorganisms. As they are very active, so they can move from one place to another place, and in this way they can carry microorganisms along with them, and in this way they find out new places, not only for themselves but also for microorganisms. Further, in ecological importance, one important fact is that nematodes are generally less abundant than protozoans, but they have an equal contribution to protozoans in case of nitrogen mineralization. So in this way, they have a very important role in nitrogen mineralization and nitrogen cycle in the soil. The nematodes increase in numbers more on materials with low CN ratios. Low CN ratios means low carbon but high nitrogen. I mean low carbon to nitrogen ratio. So such materials contain more nitrogen. So these nematodes prefer material with more that contain more nitrogen in it. Therefore, they play an important role in nitrogen mineralization process. And that may be why when there is an increased input of nitrogen in soil, the number of nematodes also increase. And another thing is that when there is more plant cover, when there is more primary productivity of a region, the number of nematodes also increase. So it means plant cover also increase the number. The soil structure has a very important impact on nematodes. As nematodes increase in soil, nematode population is higher in the soil with higher porosity. For example, in sandy soils are more porous, so they have more nematodes. The reason why the nematodes are higher in soil with higher porosity is simply because of pore spaces, because they can only move through these pore spaces. They cannot change their shapes according to the pore spaces, so they need space to move. It's unlike the protozoans, which can modify their shapes and move, but for nematodes, they need the space according to their body. And another important fact that has been observed is that nematodes decrease with increase in soil depth. And this fact can also be related to the availability of food material. And then if we compare the number of nematodes in different environments based on their plant cover, we see that highest nematode highest numbers have been reported in forests, followed by meadows and then by prairies. So we see that with the change in the ecosystem, in the type of plant cover, the type of nematodes there also increase. For example, in woodlands, it has been observed that the nematodes which feed on bacteria are more in numbers, while in grasslands, nematodes that eat plants, they are more in numbers. So another important character is that the nematodes are affected by the cultural practices. Cultural practices mean how you manipulate the soil for agricultural activity, how you or how many, or what type of cultivation practice you do, how you plow the soil, etc. It has been observed that with increased tillage, the fungus feeders increase in population. So this may be because of the reason that when we increase tillage, we promote aeration of the soil. And in this way, fungus increase, fungal attack increase on the available organic matter. And with increase in fungal population, nematodes which feed on fungus, they also increase. Then integrated management practices has been observed to increase the microbe eaters. And the organic manure application also increased the nematode population. This may be related to the increase in organic matter contents of the soil. When we apply more organic matter in the soil, it will increase more detritus eaters. And then the irrigation also increases the number of nematodes. This may be related to the plantation, plant cover, and increased organic matter due to availability of moisture. Then it has also been observed that with changes in the weather or with seasonal fluctuation, the numbers also change. So how the nematodes are distributed? So based on the available literature, it has been observed that the resources available in a place is the highest or is the most important factor that control the number and type of nematode population there. So another important thing that as nematodes can actively move from one place to another place, so their population can change abruptly. The change in population is because of two important reasons. One is emigration of nematodes from one site to another site, and other is the death of nematodes. The emigration leads to inoculation of new sources. Emigration is because of the conditions, or because of some unfavorable conditions that develop in one area, and the nematodes then move from that place and find some other place where they can live and feed. So, if we compare the plant rhizosphere and the soil that is away from the rhizosphere, we see that the nematodes are 70 times more in the area that is called rhizosphere, while as compared to the surrounding areas. It's 70 times more in numbers in the rhizosphere as compared to the surrounding areas. So these nematodes are higher in this area because of high availability of nutrients. And another thing is that the type of nematode population available in the rhizosphere or the type that is called known as plant feeder. So as we see that nematodes respond to environment very quickly, there is a prompt reaction of nematodes towards environment, and they readily move or they change their shape or something. I mean, they change their behavior. So that's why they can also be used as environmental indicator. As we discussed in our lecture in which we talked about protozoans. Protozoans are also used as environmental indicator, and in the same way nematodes can also use. Now we talk about the extraction or identification of nematodes. The extraction of nematodes from soil depend on their ability to wriggle through a mesh. So if you see this picture, here you see a mesh, and here you place the soil. Soil is placed in the form of a slurry. Slurry is prepared in water. Then it is placed on the mesh here. The active nematodes can move through the mesh while large amount of soil are maintained on the mesh. The nematodes, along with water, come down into the funnel. Here you see there is a small hose, or you can say a pipe kind of thing, with a clamp at the end. So the active nematodes lie here, and whenever you need to collect nematodes, you just open this clamp a little bit, and nematode along the water will move down in this beaker. This, and from here you then process it further for your experiments. These nematodes can be counted and then studied under the stethoscopes for identification, etc. Then, apart from this molecular method, apart from this morphological or phenotypic method, there are some molecular methods for nematode estimation. In this case, nucleic acid can be used for characterization of nematode. Similarly, RNA extraction is used for active genetic profiling, and then biomass can be assessed visually through using an eyepiece. Now, another important thing that we discuss when we talk about soil nematodes. The soil nematodes are very important also because they cause considerable amount of losses in plants by causing different kinds of diseases. So the different plants damaged by nematodes include all kinds of plants, ranging from the foliage plants through vegetables, fruits, nut trees, tough grass, to forest trees. There are different types of nematodes that cause different kinds of symptoms in the soil. For example, root knot nematode that makes knots on roots. This is a very important disease in vegetables and many other plants. Similarly, the cyst nematodes, which makes cysts on the plant body. Then there are a number of other diseases caused by different kinds of nematodes, including root lesion, spiral burrowing, bulb and stem, reniform, dragger, bud leaf, mine wilt disease, etc. So you see that different types of nematodes cause different types of symptoms in plants. So this is all about the presence of nematodes and a little bit about their different characters. So if you have any question, so please share with me. So I will respond to you accordingly on KCMS. Thank you very much.